Welcome to Chapter 9, which is all about inheritance. So this is going to be a lot of information on Mendelian genetics. So when we want to talk about Mendelian genetics, there's some things we're going to want to go over. So the first is the idea of a gene. A gene determines an organism's specific traits. So a gene is located on a chromosome. A single chromosome can have many genes. So diploid organisms have a pair of each chromosome, as we've established. So how many genes for a single trait does an organism have? Yes, correct, 2. So you have 2 copies of every gene in your body, in your cell. Every single cell, except a sex cell, has 2 copies of every single gene. Gregor Mendel, a man who determined the patterns of inheritance first, didn't know what a gene was. He didn't know what DNA was. He didn't know what genetic information was. So when you think gene, and you're talking about Mendelian genetics, allele is the same term. So an allele is synonymous with a gene. So when we're prepared to talk about Mendelian inheritance, let's talk about the differences between traits and characters. A character is a category, right? So the color of a flower, the color of hair, cats having a specific fur pattern, or height, those are all categories, whereas a trait is the specific variation in a single category. So they're variants of that character. So when we have the character flower color, there might be 2 separate traits you see, purple or white. Both are traits of the character flower color. So let's play here. The man is 6'1" and has green eyes. How many characters are in this sentence? What's that? Okay, I hear eye color. That is, in fact, one of the characters. There it is. Height. Yep. 1 more to go. And there it is. Gender. Congratulations, you guys. You're geniuses. Throwing out the correct answer on me. So the characters are gender, height, and eye color. And the specific traits are male for gender, 6'1" for height, and green for eye color. Simple stuff. So now we're going to talk about inheritance, which is what you get when your rich uncle dies. Okay, well, maybe not so much that. In biology, the term inheritance is when genes or alleles are passed on to you from your parents. So you have 2 alleles for every trait. And then when we talk about what traits you show, it's going to be important to use the term gene expression. So genes are expressed. When a gene is expressed, you see it. So considering you have 2 genes for every trait, 2 alleles for every trait, it doesn't mean both alleles are automatically expressed. So gene expression is governed by dominance, in that an allele can be dominant or recessive. And a dominant allele or a gene is always expressed. If one of your 2 alleles is dominant, you show the dominant trait. So a recessive allele is completely obscured by a dominant allele. When you have a dominant and a recessive allele as your 2 genes, the dominant is the one that's expressed. The recessive is ignored. You only show the recessive trait, you only express a recessive allele or a recessive gene if both alleles are recessive. both of your two copies of alleles for that trait are recessive. Okay, next up is the concept of zygosity. Zygosity refers to some vocabulary for when we want to talk about the combinations of genes you show, because we know that you have two alleles or two genes for every trait. And so we're going to need some words to describe whether or not those genes are dominant or recessive. So, homozygous is a term we use when we want to refer to an organism as having two of the same allele. So, if you have two dominant alleles for a trait, you're homozygous. If you have two recessive alleles for the trait, you're homozygous. Homo means same. So, both genes are the same. You can be homozygous dominant, implying that both alleles are dominant, or correct, homozygous recessive. That was an easy one. All right, if homozygous is the same, because homo is the prefix for the same, then what is the prefix for when the alleles are different? Did I hear someone say hetero? That's correct. Heterozygous is the other possible category. And in a heterozygous situation, one of the alleles is dominant and one of the alleles is recessive. So, pretty simple. One dominant allele, one recessive allele. All right. Now, next up is the concept of the true breeding organism, in which something that is referred to as true breeding, no matter how many times you cross true breeding individuals with the same trait, they always produce the same trait. So if you're talking about a plant that has a purple flower, if it's true breeding purple, anytime you cross a true breeding purple with a true breeding purple, you get purple, no matter how many times you cross it. And anytime you cross a plant that has white flower color, true breeding white times true breeding white always produces white flowers. True breeding. So, true breeding, there we go. True breeding white times true breeding white is a true breeding white. These are homozygous individuals, when you think about it, because a parent passes on their own genes. So if we have a true breeding purple, it has to be homozygous for purple color. And so it's going to pass on the purple gene. Breed it with another true breeding purple that passes on the purple gene, then both alleles are for the purple flower color. So homozygous individuals, when you breed two homozygous individuals, you're breeding true breeding organisms. So Mendel, Gregor Mendel, lived from 1822 to 1844, and he was an Augustinian friar. And he really, really, really loved him some pea plants. Like, seriously loved pea plants. How much did he love gardening, you ask? Well, he grew 29,000 pea plants. He grew and recorded data on 29,000 pea plants. It's kind of ridiculous. I feel good when I grow 3 tomatoes over 2 years. And this man grew 29,000 pea plants. Needless to say, gardening was his thing. So why peas? Well, peas have some very predictable traits. Gregor Mendel was able to look at the traits of flower color, plant height, seed color, and seed texture. And they were all very predictable and easy to examine, which is useful because it was generally frowned upon at that time to try and force people with certain traits to breed, and then force those offspring to breed in a certain way. Not only was that sort of frowned upon, but, you know, your time between generations, 16, 18 years, not that easy to pull off. Pea plants breed very quickly. You can get 2 generations over a course of a couple of months. So, ideal for breeding massive amounts and examining traits. And through breeding his pea plants, he discovered the rules that govern Mendelian inheritance, Mendelian genetics. So let's look at breeding peas. Mendel looked at flower color, and he used the letter P to abbreviate the alleles for flower color. A capital P represented the dominant purple, whereas a lowercase p would represent the recessive white allele. So the dominant allele was purple, represented by a capital P. The recessive allele was white flowers, represented by a lowercase p. And then seed color with the abbreviation Y. So yellow is the dominant trait, capital Y. Green is the recessive trait, lowercase y. Height, tall versus dwarf. Tall is dominant, capital T. Dwarf is recessive, lowercase t. And finally, seed texture. Round is dominant, capital R. Wrinkled is recessive, lowercase r. So he assigned each trait an allele. An allele was his term for the genetic unit, what we now think of as a gene. Each allele codes for a single trait. So a genotype was a term he thought up for the sum of alleles. So both alleles for a given trait represent the organism's genotype. So the genotype for flower color would be 2 alleles for flower color. So maybe heterozygous, or homozygous dominant, or homozygous recessive. That's the genotype, which can be contrasted with the phenotype. The phenotype is the physical appearance. In other words, what is expressed, what it looks like. So why have genotype with separated from phenotype? Well, what does a homozygous dominant flower color plant, pea plant, look like? What color is its flowers when it's homozygous dominant? Did you say purple? Ah, good for you for listening. How about the flower color for a heterozygous plant? Correct, also purple. So a homozygous dominant and a heterozygous plant both have the same phenotype, purple flowers. However, they have different genotypes. So, if you're breeding peas for flower color, your options are purple or white. A true breeding purple times a true breeding white plant, what Mendel referred to as his parental cross, always produced the phenotype purple flowers. Always. So, well, which one is dominant and which one is recessive? Well, we had purple and we bred with white, and they were all purple. So purple is covering up the white. So we know that our offspring ended up with 1 allele from one parent, the purple parent, and 1 allele from the other parent, the white parent. So the offspring all had 1 dominant allele and 1 recessive allele, because a true breeding plant is either homozygous dominant or homozygous recessive. So our offspring had 1 dominant and 1 recessive allele. So what kind of zygosity does it have? Yes, heterozygous. They are all heterozygous. So they were all purple. So all the offspring of our parental cross of a true breeding purple times a true breeding white have the phenotype purple, and they all have the genotype heterozygous. But then Mendel went one step further and crossed 2 of the heterozygous offspring from that original parental cross. He referred to this as the F1 cross. And in the F1 cross, most of the offspring were purple, but some were white. About 75% of the offspring were purple, 25% were white. How exactly did this happen? Well, purple is dominant, so we have the capital P. White is recessive with the lowercase p. A true breeding purple is homozygous dominant. A true breeding white is homozygous recessive. So hybridization is when two different true breeding plants are crossed. So flower color. Here is a heterozygote. The character is flower color, as I just said, and the trait is, that's right, purple. The zygosity is, yes, heterozygous. The phenotype is, correct, purple. The genotype, yes, big P, little p, heterozygous. So a lot of this is repetitive, but it's good to know the differences between all of them. So when we go to explain why crossing two heterozygotes produce purple and white flowers, we need to talk about the law of segregation. So when I say one parent donates one allele and the other parent donates the other, it's important to know that the parents donate one of their two alleles. So the law of segregation says the gametes get one of the parent's allele from every allele pair. So if my parent is heterozygous, the gamete will get either the dominant allele or the recessive allele. Not both, because it's a gamete and it's haploid, only has one chromosome. So it will only end up with one of the parent's alleles. So it can either be dominant or recessive in a heterozygote. So let's take a look here. If mom is heterozygous for trait or character represented by P and the character represented by F, what possible genotypes can mom's eggs show? Well, each of her gametes will get one of the character represented by P and one allele from the character represented by F. So she could be capital P, capital F, capital P, lowercase f, lowercase p, capital F, and lowercase p, lowercase f. Let's take it a step further. Here's another one. Well, this is kind of deceptive. We have 1, 2, 3, 4 traits going into this gamete. But if we take another look, the gamete can only get a dominant trait from this pair, and it can only get the dominant trait from this pair. So again, we're going to have a situation where there's only 4 different possibilities, because the only ones where we're going to have variation are these T's and G's, and they're heterozygous. Okay, so it can get more complex from here. You can have 3 traits that are all heterozygous, 4 traits that are all heterozygous, all the way up through the haploid number, 23. So, you can have a gamete with 23 possible, alleles. And remember, 2 to the 23rd, that's 8.4 million combinations in a human. Ridiculous. So if we have 2 traits, here we go, flower color and seed color. Purple and yellow are the phenotypes here. All the possible gametes from mom, well, she's homozygous dominant for flower color and heterozygous for seed color. So she could have capital P, capital Y, or capital P, lowercase y. That was easy. Let's go ahead and do something more complex. Here's mom when she's heterozygous for both flower color and seed color. So now she can get all 4 possible combinations. Dominant allele for flower color, dominant allele for seed color, so purple, yellow. Dominant allele for flower color, recessive allele for seed color, purple, green. Recessive allele for flower color, dominant allele for seed color, white, yellow. And then recessive allele for flower color, recessive allele for seed color, white, green. And here's dad. He's also heterozygous. So let's do a little basis here. We use true breeding plants for our initial parental cross. And so our phenotypes are purple flowers crossed with white flowers. And when you cross them, because each was homozygous, our purple parent was homozygous dominant, our white parent was homozygous recessive. Purple parent can only donate a dominant allele. The white parent can only donate a recessive allele. So they all are heterozygous. F1 is heterozygous. So now we cross our F1 offspring, two heterozygotes. Do they all have the same genotype? No, they don't. How do we know? Well, let's take a look. So, a little bit of vocab before we bust into there. The concept of generations. The parental generation, or the P generation, are the first plants you started with. So in Mendel's case, the true breeding plants. And then the F1 are the offspring of the P generation, the first round of offspring. And the F2 are the offspring of the F1. And it can go on from there, F3, F4, F5, just describing how far back from the P generation you are. So let's make some kids. Our first cross is the parental generation. Homozygous dominant times homozygous recessive. What genotype is mom and dad versus dad? Well, doesn't really matter because either one of these could be the female or male parent. So what is the only allele that mom can donate? Let's call mom the homozygous dominant. She can only donate a dominant allele. Because both her alleles are dominant, doesn't matter which one of those two alleles she randomly gets, it's dominant. And then with dad being homozygous recessive, both his alleles are recessive, so it doesn't matter which random one of those two he gets, it's recessive. So all of the offspring are, once more with feeling, heterozygous. So now the heterozygous cross is a little more complicated, because in this case, our heterozygote could donate randomly to their gamete either a dominant allele or a recessive allele. And in order to determine the possibilities we can get from crossing two heterozygotes, we need to do a Punnet Square. So what is a Punnet Square? Well, this is awesome! Time to get excited, because Punnet Squares are cool. So here's the basic setup of a Punnet Square for crossing a single character, flower color in this case. Mom's genotype, she's a heterozygote. Dad's genotype, heterozygote. So first thing we want to set up are mom's gametes. She can donate a dominant allele to her gamete, or she could donate a recessive allele to her gamete. Then dad, he can donate a dominant allele or a recessive allele. Dominant, recessive, dominant, recessive. So this represents one of the two possible alleles he can donate. So now we do the Punnet Square, and we start putting together the alleles when dad's sperm fertilizes mom's egg. So it's random fertilization, so dad's sperm carrying this dominant allele could fertilize mom's egg carrying a dominant allele, in which case we get homozygous dominant, or we could have dad's recessive sperm fertilize mom's egg carrying a dominant allele, in which case we have a heterozygote. Then we get the dad's sperm with the dominant allele and mom's egg with a recessive allele, another heterozygote. And then finally we have dad's sperm with a recessive allele and mom's egg with a recessive allele, giving us a homozygous recessive. These are the 4 possible combinations of gametes we can get. So our phenotypes, we have 3 possibilities here that are purple because they carry the dominant allele. Doesn't matter that this one's homozygous dominant and this one's heterozygous. They both have a dominant allele, so they're both purple. So 3 possibilities for purple flowers, 1 possibility for white flowers. And then when we look at the genotypes, we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. If we were to express this as a ratio, we'd have 3:1 for a ratio of 3 purple flowers for every white, 3:1. And then the genotypes, very much the same, 1:2:1, or 1:2:1, as far as for all, 1 out of every 4 genotypes is homozygous dominant, 2 out of every 4 is heterozygous, 1 out of every 4 is homozygous recessive, 1:2:1, or 3:1. So this example was a monohybrid cross, in which you cross for a single characteristic. A single characteristic, like flower color, like seed color, seed texture, plant height. You cross for a single characteristic, that's a monohybrid cross. That's your very simple 4-square Punnet square. But there's also the dihybrid cross, in which we cross for 2 characters at the same time, which makes sense because a plant doesn't only have a single character, it's going to have all these characters at once. You can make it even more complex with a trihybrid cross and like a tetrahybrid cross or a quaternary hybrid cross. I don't know what the term would be. But a trihybrid cross would be 3 characteristics, and what the heck we'll go with tetrahybrid cross would be 4 characteristics. It gets very complex. But the extent of our complexity is going to peter out at dihybrid crosses. So we're setting up a dihybrid cross. Mom's genotype is heterozygous for both traits, flower color and seed color. Dad's genotype, again, heterozygous for both traits. We cannot separate these out and make just a single Punnet square for flower color separate from seed color, because these are inherited together. So, figuring out our gametes. In a dihybrid cross, the gamete has 1 allele for each character. So our gamete is going to have 1 of the 2 possible alleles for flower color and 1 of the 2 possible alleles for seed color. So here's Mom. She's heterozygous for both. Let's see what our possible combinations are. Well, there we go. She could have the dominant allele for flower color and the dominant allele for seed color. Or she could have the dominant allele for flower color and the recessive allele for seed color. Or she could have the recessive allele for flower color and the dominant allele for seed color, and then finally the recessive allele for flower and seed color. These are the 4 possible gametes that we can have produced by a single parent. So here we go, Mom's gametes. There we are, all those 4 possibilities. And you can see 4 squares in the row. And since Dad is also heterozygous, we're going to have the same possibilities. So now let's do this sucker. Here we go. This is homozygous dominant. Next up, homozygous dominant for flower color, heterozygous for seed color. Then heterozygous for flower color, homozygous dominant for seed color. Heterozygous for both flower and seed color. Homozygous dominant for flower color, heterozygous for seed color. Homozygous dominant for flower color, homozygous recessive for seed color. Heterozygous for both. Heterozygous for flower color, homozygous recessive for seed color. Heterozygous for flower color, homozygous dominant for seed color. Heterozygous for both. Homozygous recessive for flower color, homozygous dominant for seed color. Homozygous recessive for flower color, heterozygous for seed color. Almost through. Heterozygous for both. Heterozygous for flower color, homozygous recessive for seed color. Homozygous recessive for flower color, heterozygous for seed color. And finally, our lone homozygous recessive for both. So here we go. Let's count up the phenotypes. We can have purple and yellow, purple flower colors, yellow seeds. We could have purple flower colors, green seeds, white flower colors, yellow seeds, and white flower colors and green seeds. So purple's dominant, yellow's dominant. So here's 1 purple and yellow. Here's 2 purple and yellow, 3 purple and yellow. Getting pretty easy. 4 purple and yellow. 5 purple and yellow. Oh, there's a purple green. So 1. Oh, there's another purple yellow. So now we're at 6 to 1. Oh, 6 to 2. 7 to 2. 8 to 2. Oh, 8 to 2 to 1. There's our first white flower color, yellow seed color. And then there's our second white flower color, yellow seed color. Here's yet another purple yellow. And that's going to be the last possible purple yellow we have. So 9 purple yellow. Then there's a purple green, so there's 3. There's a purple yellow, or a white yellow, so there's 3. Leaving finally our little homozygous recessive for both, 1. Which makes our ratio for phenotypes in a dihybrid cross in which both parents are heterozygous, 9 to 3 to 3 to 1. Which makes sense because if you add up 9 plus 3 plus 3 plus 1, you get 16, and there were 16 possible combinations of gametes. The genotypes, oh no, that's a lot. If anything, you could get extra credit on an exam for writing out the genotypic ratio in this sucker. Otherwise, it's pretty darn crazy. So we're not going to deal with genotypic ratio as far as what you're required to know on an exam. However, there may just be a genotypic ratio question on the exam for extra credit. So ratios, I guess we'll pop through this and then end this particular batch of the recording. We could have phenotypic ratios and genotypic ratios. How do we calculate them? The number of specific phenotypes possible divided by the total number of possible phenotypes is the percentage chance of it occurring. So then you reduce. And then the same thing for genotypes. So let's look. Here's true breeding parental generation. And our F1 is 4 heterozygotes. So we go for the F2 by crossing 2 of our heterozygotes. What's our genotypic ratio and our phenotypic ratio? Well, how many possible purple were there in this cross? 3. Correct, 3. And how many possible white? 1. That's right, 1. So our phenotypic ratio is 3 to 1, and our genotypic ratio was 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. So 1 to 2 to 1. 1 to 2 to 1 genotypic ratio, 3 to 1 phenotypic ratio. So for a monohybrid cross with heterozygotes, the following is also always true. You always get a 3 to 1 phenotypic ratio if we're heterozygous for our trait, no matter the trait or character. And it's always a 1 to 2 to 1 genotypic ratio, no matter what character you're looking at. If it's a monohybrid cross with heterozygous, phenotypic ratio is 3 to 1, genotypic ratio is 1 to 2 to 1. How about a dihybrid cross? Well, here's our true breeding parental generation, getting our F1 of heterozygous for both, crossing the F1s, producing an F2 that is, well, we just did it, always a 9 to 3 to 3 to 1 ratio. If both parents are heterozygous for both characters, the resulting phenotypic ratio is always, always, always 9 to 3 to 3 to 1. Whether it's flower color and plant height and seed color, plant height and seed texture, if you're heterozygous for both, always 9 to 3 to 3 to 1. Easy. Then we calculate the probability. Well, let's calculate the percent chance of having a specific phenotype or genotype. In a monohybrid cross, 3 to 1 phenotypic ratio. Well, 3 to 1 is 3/4 to 1/4, because there were 4 possibilities. 3/4 is 0.75 times 100, 75% chance of the offspring being purple. 1/4, 25% chance of the offspring being white. Dihybrid crosses, pretty much easy, except in this case, instead of 3/4, our denominator is 16. So, pretty much the same thing, 9 to 3 to 3 to 1. So our chance of having purple flowers and yellow seeds is 9/16 times 100. Purple flowers, green seeds, 3/16 times 100. White flowers, yellow seeds, 3/16 times 100. White flowers, green seeds, 1/16 times 100. So 56.25% chance of purple yellow, 18.75% chance of purple green or white yellow, because they're both 3/16, and a 6.25% chance of having a white flower with a green seed. Easy, easy, easy. So use your ratios to determine the percentage chances of genotypes or flower color. So probability of a white flower is correct, 25%, because this is a heterozygous cross. So it's always 3 to 1, 75% to 25%, since the white flower is recessive. Probability of a purple flower, bam, just said that, 75%. Why are the probabilities random? Why is it like flipping a coin where it's a 50-50? Why do we express it as a percentage chance? Well, remember independent assortment, where it's a 50-50 shot whether or not the gamete gets one of the two alleles or chromosomes or genes from the parent? Well, bam, that introduces randomness. So which allele the gamete gets out of every allele pair is completely random. Each pair of allele segregates independently of the others. So with PPYY, flower color, seed color, a recessive P doesn't always segregate with a recessive Y. You can get a recessive P with a dominant Y. So there we go. It could be this or this in our gamete. Remember, the gamete has only one allele of each character. So bam. The alleles separate independently in the law of independent assortment. We'll kick back with how to figure out genotype if you don't know it.